Sure. Thank you. Thanks. Good morning, everyone, again. And again, my apologies for not being able to attend this meeting in person. Uh, I had a terrible experience yesterday at the airport, and eventually my flight got canceled. And thanks for the opportunity to present remotely this morning. My name is Volkan Yesilyurt. I am a senior scientist in Takeda. I am part of the drug product and device development group. I am particularly in charge of the new modalities, drug delivery type of formulation development. Today, I will talk about lipid nanoparticle formulation development and their applications beyond vaccines. I want to very briefly introduce our organization. So we are CMC. We have three major locations. Our location in Lexington, mainly responsible for biologics formulation, device development, and combination products. I am located in Cambridge, and together with the Japan site, my group is responsible for small synthetic molecule formulation development. So we pretty much cover everything from small molecules to oligonucleotides, sRNAs, ASOs, everything synthetic. Globally, we are around 80 people. And this is the outcome of my presentation this morning. At the beginning, I want to briefly talk about the nucleic acids, their similarities, major differences compared to traditional modalities. I will continue with the issues that affect the clinical translation of these nucleic acids. Here I will highlight the strategies that help to overcome these challenges that include chemical modifications, conjugations, and particle-based approaches to tackle these delivery and stability challenges. So here the main focus will be on the LNPs, lipid nanoparticles. I'll talk about their targeting capabilities, pros and cons, and I will conclude my presentation with the strategies to aim LNPs at organs other than liver and their applications beyond vaccine. So here I am showing you some cartoon representation of the most common nucleic acids. So these molecules, they have great potential to target genes and gene products. For example, mRNA can be used to replace a missing protein or enzyme. With sRNA, we can silence, knock down any disease-causing proteins enzyme. With CRISPR, it has become very popular in the last 7-8 years. We can permanently edit correct genes. So when you look at them, they are very different in size. ASOs and sRNAs, they are much smaller compared to mRNA, plasmid DNA, and CRISPR systems, and that affect their, the formulations, the other things. And since sRNA and ASOs, they are much smaller compared to other nucleic acids, we have the ability to chemically synthesize them. So we can modify them, we can do conjugations to not only improve their stability, but also facilitate their cellular uptake and get better potency. And in contrast to these traditional modalities, again, these molecules, they need to go inside the cells. They need to go into the cytoplasm or nucleus to perform their job, and there are multiple barriers, right? After the IV administration, so we have multiple extracellular barriers. We have macrophage clearance, kidney clearance of these nucleic acids. We have so many enzymes, nucleases in the extracellular compartment that quickly degrades these nucleic acids. So their half-life upon administration is usually less than 10 minutes. And if you look at these molecules carefully, they are, they are highly charged. They are negatively charged. They have a very large size, and and they are hydrophilic in nature. So our cells, they have all the reasons not to allow these molecules to enter into the cells. And that's mainly because of their physicochemical properties. And even if somehow we can send these therapies inside the cells, we have other barriers there. So they remain trapped inside the endosome, and they need to escape from endosome and release into the cytosol to function. So that's another additional barrier for these modalities to pass to fully access their potential. So then the question is, so I mean, how we can overcome these challenges? So what has been done not only protect these modalities from degradation and but also to facilitate their cellular uptake, endosomal escape, and then the later efficient encapsulation in cytosol. So I will discuss this in more details in the following slides, but there are mainly two approaches to tackle these important challenges. So the first approach is the chemistry, so chemical modifications, conjugations, increases their stability, circulation, half-life, and their targeting capability. But remember, I just mentioned you in the previous slide that the chemical modifications can be only applied to small oligonucleotides, sRNAs, ASOs, but we have other cargoes like mRNA, CRISPR, they are very large and they are biologics. So we cannot use chemical strategies to improve their stability and other problems. So usually we go with a particle-based approach, whether it's a viral or non-viral, to protect these larger cargoes from the extracellular barriers and also to facilitate their cellular internalization and then later the translation or expression, whatever these cargoes are designed for. So let me tell you a little bit about the delivery strategies that involve chemistry. So if you look at this one example of a nucleic acid here, they have backbone based on the phosphate esters, we have the sugar units, we have the base units. So we can chemically modify all these sites to improve their stability and potency. And in fact, actually, if you look at the most of the approved ASOs and sRNAs and many in development, they all involve modification to their phosphate group. So these phosphate groups, they are very susceptible to a rapid degradation by the enzyme. So changing this oxygen to a sulfur atom makes them really resistant to nucleases. So we have the ability to increase their blood circulation half-life. In addition to this backbone, this phosphate modification, the other common modification involves the attachment of different groups to these two prime groups. So usually people use, fluorine atom or methyl other groups to make these molecules little hydrophobic to promote their cellular uptake. So, and other than these two common approaches, I mean, there are more advanced type difficult modifications that involve replacement of this sugar with another sugar moiety or using modified nucleotides, again, mainly to increase their stability and the potency. But again, these chemical modifications itself, they are not always enough to promote cellular uptake. So they are mainly to improve their stability. With ASOs, they are very small, around 5-6 kilodalton molecular weight. So they are heavily chemically modified, and they don't need any targeting moiety conjugation or any delivery agent, actually. So these molecules, they have the ability to enter into the cells, then go into the nucleus. So we don't know exactly what the mechanism is, but ASOs, once we improve their stability through chemical modifications, they can penetrate through the cellular bilayer and reach the nucleus. But we cannot do the same thing for sRNA. So sRNA, 2 to 3 times bigger than ASO and much smaller than mRNAs. We can chemically modify them to increase their stability, but that does not improve their cellular uptake. So to improve their cellular uptake, people have come up with an idea of attaching, targeting moieties to these sRNAs to increase their ability for cellular uptake. So here you see so this is the one of the approved products where 3 GalNAc moiety conjugated to sRNA. And the mechanism here is that the liver hepatocytes has many glycoprotein receptors. So these GalNAcs can bind to those receptors to facilitate the cellular uptake of this sRNA itself. Otherwise, it is very difficult to get into the cells without having this GalNAc groups conjugated to it. And another advantage of these conjugates, they don't need a delivery agent, right? When you do nanoparticle-based formulation, so you have multiple components, you have CMC challenges, stability issues, but with the conjugates, you don't need all those formulations to deliver these therapies inside the cells, because the GalNAc conjugated sRNA is itself enough to have a better circulation and to better penetrate inside the cells. But again, I mean, all these modifications that I just mentioned, the modifications to sRNA, conjugations to sRNA, or chemistries for ASO, they're good for small cargoes. So, but we have these larger cargoes, right? mRNA, DNA, CRISPR. So we need to protect them. So we need to put them in a protective layer to increase their stability against the enzymes during the circulation, and also to promote their cellular uptake and the translation later. I mean, I'm not going to talk about the virals in viral-based vectors today, but what I can tell you here, when it comes to gene delivery to the nucleus, the virals so far the best vectors which can achieve the DNA delivery into the nucleus. And that's mainly because this is what they are designed for. So they can penetrate through the cellular layers, they can go inside the nucleus, and they can incorporate and release their cargo into the nucleus. But with the particle-based approaches, when you have the gene, it does with DNA, it is very difficult to take this cargo into the nucleus. We have much better chance with the mRNA and other cargos, because these molecules, they don't need to go into the nucleus. So we just need to release them into the cytoplasm, where they do the cellular machinery to express proteins or to silence proteins. So that's the biggest advantage of the viral compared to non-viral, because of its ability to take genes into the nucleus. And we have multiple products by Modalities, Sport Therapeutics, which involves viral-based delivery for genes for different applications, such as SMA. Okay, so we have the larger cargos, and the only option, if you want to go with the non-viral, particle-based approaches. There are a wide range of biocompatible materials which are designed to complex, encapsulate, and deliver this nucleic acid. Among them, LNPs represent the most advanced platform for systemic delivery. If you look at here, this LNP is usually composed of 4 lipids. We have a cholesterol, we have a PEG, we have a helper lipid, And we have the ionizable lipid, which is proprietary most of the time. In this case, it's MCT. It is the approved lipid used in FDA-approved product such as Onpattro. PEG lipids important because it increases the blood circulation. It reduces the particle aggregation, and other than PEG lipids, cholesterol and helper lipids, they are crucial because they increase the stability of the lipid bilayers. When it comes to LNPs, I mean, you always want to go with a smaller size. Their size usually ranges from 50 nanometer to 120, 130 nanometer. You want your LNP to efficiently encapsulate your cargo, whether it's a small cargo like sRNA or it's a very large cargo like mRNA. We know that we need a really low surface charge, important to avoid the immune recognition and any unwanted side effects. And the pK of the LNP has been shown to be really important. pKa is around 66.5, is crucial for efficient endosomal escape, which I will talk about it in a minute. So I just told you one of the key components of the LNPs is the ionizable lipids. So these lipids play an important role in the delivery efficacy of LNP formulations. So they have ionizable head groups, which is protonated during the formulation so that it can complex with the negatively charged cargos. It is neutral during the blood circulation to avoid any systemic toxicity and any interactions with the serum proteins. And once it is inside the cells, and the pH in the endosome is acidic, so this ionizable lipids can get protonated, and that's how it helps to escape the endosome. So that's why these are really key lipids in the formulations. MCT is the first one approved. We have Moderna lipid and the Pfizer lipid used in the vaccine formulations. And one of the common features of these lipids, they're all, as you can see, biodegradable. So you want your lipids to degrade to non-toxic byproducts, especially for repeating those, like let's say Onpattro, it's always, you don't want accumulation of your LNPs lipids at the liver, so you want it to degrade quickly into the non-toxic byproducts. This is another lipid from my postdoc lab, Langer and Dan Anderson. It's a very efficient non-degradable lipid for used for both sRNA and mRNA. But the one thing important about these lipids, they are proprietary. You either buy them, license them, or you make your own lipid. Like the Moderna, they use their own ionizable lipid. Pfizer, they license from Acuitas. So what is important about these lipids, their CMC, their impurity profiles, their in-use stability, storage stability, these are very important for clinical translation. Okay, so once you have your lipids, ionizable lipids, and other components, the LNPs can be prepared by rapid mixing of the ethanol with nucleic acid cargos in acidic buffer using a microfluidic mixing. In our labs, we have these two mixing devices. For small scale, we use this can make LNPs up to 15 ml, and the other one can go up to 1 liter. But you don't really always need these devices. Not every lab has. In our postdoc lab, we use just 2 syringe pumps, and we use in-house made PDMS mixing device to make the LNPs. So what is important about LNP formulation? The mixing speed, the buffer, the ratio of your lipids to the cargo, they all affect your formulation parameters and they need to be optimized. So once we make the LNPs, then we look at size, surface charge, encapsulation efficiency. And if you have large amount of LNPs, lipids, then it's better to do in vitro screening and do the in vivo later to how effective your LNPs are. So far we talk about these LNPs, their compositions, importance of the ionizable lipids, but what is special about these particles? How do they work in vivo? So it has been shown that the remarkable affinity of these LNPs for the liver hepatocytes is mainly because of the absorption of apolipoprotein E on the surface of these particles. So upon IV administration, people think these PEG lipids are replaced by the apoE which act as a targeting ligand for certain receptors, LDLR receptors on the hepatocytes. So these receptors are very common on hepatocytes. They have million on each cells, and this apoE LDLR receptor ligand interaction are responsible for the liver uptake of these LNPs. And people have shown that when you use the animal model with certain gene modifications where you don't have the apoE protein, then the liver targeting and is much less compared to the regular mice where we have the apoE protein in the circulation. So if I am not missing any new products so far, we have only 4 LNP products approved by the FDA, and we all know the LNP mRNAs from Pfizer and the BioNTech and the Moderna. So we have 2 more LNP products. They are based on sRNA. The first one is the Onpattro, and the other one is run by the Novartis. So other than the vaccines, but if you look at these approved products, including the GalNAc conjugates, and there are hundreds in the preclinical and clinical development, they all target liver, right? So this liver is kind of a low-hanging fruit for LNP's clinical translation. So if you do some literature search and pick some LNP-based publications, this is a very typical graph where you see the biodistribution, imaging of the organs that shows all these LNPs mainly goes to the liver, and there is negligible amount in the other organs. So, and that's because of this apoE mechanism that I just mentioned. So then the question is, are these LNPs limited to only liver and vaccines, and how we can unfold their full therapeutic potential for the other organs, right? So can we still deliver them to liver, but not the hepatocytes? We have other cells in the liver like stellate cells, which is important for liver fibrosis. Lung targeting is important because cystic fibrosis there. People start using LNPs for immunotherapy and local delivery for eye. So there is some progress, and there are some research ongoing on with the targeting ability of the LNPs to the different organs. Basically, there are 2 strategies. We call them passive and active targeting. With the active targeting, we have targeting moieties decorated to the surface of these LNPs. They can be small molecule peptides, antibodies, and they interact with certain organs, receptors on the cell surface for targeted delivery and cellular uptake. The other approach, which I like most, the passive targeting, because it's much simpler. It does not involve that much chemistry and CMC challenges. So basically here it's your formulation. So you play with your formulation, lipids, surface charge, all other things to somehow make these LNPs to go to the certain organ. So for this, there is not a really good understanding of mechanism or structure property relationship. Usually people make huge library of LNP with different lipids, and they sequence them and see where they are going, and then they try to come up with alterations of the formulation. So this is one publication from and it's from a company called Rico Therapeutics. It's a passive targeting, so they incorporated another lipid, the fifth component into their lipids to target them to the lungs, spleen, and the livers, and it's very effective. So with this, I want to summarize my presentation. So oligonucleotides, those ASOs, and maybe SRNAs, they don't really need a delivery agent. You don't need nanoparticles to deliver SRNA to the liver. So gamma conjugates works fine, and it seems it's better than the particle-based approaches. But for larger cargos, we have to make LNP or other polymeric-based formulations. Maybe I forgot to mention this during my presentation, but this is a really important factor that affects the clinical translation of the LNPs versus the conjugates. The conjugates, let's say that gamma conjugates, they are much smaller. They can be administered sub-Q. So the patients, they can do this at home. In contrast to LNP-based formulations that require IV administration in the hospital settings. And the liver is the primary target for most of the LNP so far, whatever the cargo is. But there is ongoing efforts and developments in the clinic to actively and passively send these LNPs to the different organs. And with this, thank you very much for your attention and listening, and I am happy to take any questions. Thank you.